So something like this. It's restricted, but it's going to be enough, at least for our showcase, to see that you have this preferential organization. All right. So briefly about the signal processing. So if you have the fluorescence as a function of time, you can recognize here the different peaks, but the problem is it decays in time, and this you want to do something about it to make sure that events that are like this are not detected as fully activated. So you have to rectify, so you have to subtract an abstract curve, which could be like the envelope of the lower envelope. So how do you do this? You look at the maximum, that's your segmentation of the events, and then what you do here, you can interpolate here, so we use here the segmentation to find this when it starts and when it ends, and then basically what you do here is you interpolate with just a linear. You can use also spline to get this lower envelope. You can also use other type of technique. We didn't use that at that time, such as empirical mode decomposition. The problem in the empirical mode decomposition here, you have some noise to be careful with because of the fluctuations of what you see here. So then you can again do this interpolation just on this orange curve, and once you have this, you subtract your baseline that you have found with the blue curve. So if you subtract this, everything will be rectified, and during this rectification you get this result. So you get your maximum, you have your baseline now, and you can identify or do a further segmentation according to where you cross here this black line. Okay, but at least now you don't have any more this change in time. Great. So before I explain how we can use this for reconstructing the graph, I wanted to discuss a bit of anatomical connectivity. What are they? If I show you this, this is electron microscopy, frozen tissue, everything is dead, you can label things, and you see that you have a contact between one synapse and another, between a bouton and another bouton, which create a synapse between two neurons. Then you can say, I have something here. I have vesicles, so we have to confirm that there is some vesicles. And if you say that you have vesicles, mostly the vesicles are here, and you have here this, maybe it's upside down, maybe this is the pre and this is the post, whatever. But you can say that according to this, you can add between the two neurons a quantity that measure that you have for sure an anatomical connectivity. Does that work? Nobody knows because it is frozen. But most likely it should work as a synapse. So if you have to do that by eye over billions of synapses, this is heavy work. So the idea in this direction is to be able to automatize this task by recognizing the structure here, and going into the tissue to identify them automatically. So I think in the last 5 years, there have been quite a bit of work going in that direction. So people looked at, they have this ultrastructure. Ultrastructure means you go beyond the cell's border, so you are inside. You want to identify those structures, which are called mitochondria here. So you want to see where, for example, they are. You want to quantify here those vesicles. And in the end, you want to have some label that identify basically each cells. So you put a label on each pieces that you have here, and even you can go beyond, as I said to you, because if you want to recognize a synapse, you see here the vesicles are not necessarily close enough. Although they are forming a cluster, they might not be close enough to the surface. So we can say this is not necessarily a synapse, but this is something I want to keep track. Maybe there is this feature can reproduce itself. So this is something we want to keep, although it doesn't have necessarily a clear interpretation. So you want to keep some information based on statistics, on local clustering, but clustering is not enough. You have to say, let me show you back what we have here. You have here, you see how accumulated they are to the surface, and sometimes it can be even more. So you want to identify features, to put them somewhere, to keep them, and to look if this is reproducible, and between which cells. And you're also interested in, for example, do you have a mitochondria? Each time you have this, at which distance you have this from your structure? Because if you don't have mitochondria, maybe there is not enough energy. And so what you are seeing is something that looks like a synapse, but maybe it's not functional enough. So you can go step by step. This is a 2003 paper, and I found quite a bit of going that direction, especially from what comes from the Allen Institute in Washington State. They have, I think, the institute over there, they generate quite a bit of data. I don't know exactly how involved they are in doing all the processing and the machine learning that is needed. Because you see, you can say, all right, let me recognize synapses. This is something you know. So if you know what you are looking for, it's easy. But can you discover? Can you discover structure that are a bit different? Can you discover synapses between different cells? And so on. Right? So this is something that we're interested to look at. This is one of the projects of Erwan to go more into these details and be more specific in different types of tissues. Okay, so just to clarify, this is the brain. So now people are able to color each cell. So you know you have each cell. You have different. This is different types of brains. So you can have different density. You can have. You see here the soma, the cell bodies, and you're interested in, for example, how many synapses has this one with that one, and you can take two neurons and ask, you know, how many synapses they have. Can you count them? Can you see where they are? Are they belonging to a specific path? And if they belong to a specific path, you want to count how many neurons belong to that path. So you start to quantify, for example, if there is some kind of column organization that could be revealed anatomically. Yeah, so finally, this is some years ago. So this is, again, electron microscopy data. So people are able, for example, to extract from this type of images the 3D, this is a 3D stack. You can take some of slices. In this neuropil, you see here that you have a dendrite with what's called dendritic spine, and this red part is called postsynaptic density. This is where you have an ensemble of molecules of interest that contribute to synaptic transmission that are positioned. And so you can play with, they have softwares that you can remove, for example, the green and keep the location of these postsynaptic densities. And again, you are interested, so this has been descriptive, right? But the question remains today, how much we can infer about the typical connectivity between neurons, the statistics, find paths, find cluster of neurons, cluster of neurons that could be involved in processing information. Yeah, so for example, I found also this paper, which is, again, the same type of ideas. You have here, obviously, vesicles. Opposed here, you have the postsynaptic density that you can recognize. So here you have obviously a contact, and you can again move on by characterizing here the different organelles. This is mitochondria, and you are interested in, from those images, for example, count, the synapses. So if you want to do some kind of machine learning approach, you would say, all right, if I have vesicles, it's not that the synapse is big like this, although you can have clustering. So I have some cluster, that's true, but to identify the synapse, you need the cluster, you need the membrane, and you need this black, dense postsynaptic density. Okay? So they developed some kind of approach for not necessarily frozen images. This is called live tissue using STED. STED is a specific type of microscopy, and they have started to look at this neuropil in 3D, look at the structure that they can get, and there is no quantification, there is no machine learning to recognize things, but it means that they have moved the technology to tissue, not frozen tissue. Okay, so this is anatomical connectivity. I think it's clear what we're talking about, that when there is a connection, you want to recognize them, you want to quantify them, you want to look at the statistics, the number of mitochondria around, the number of vesicles participating, the size here. You can look at if there are microglia cells around. You have to do all this this labeling to do this. There is possibly enough data now that has been generated, for sure. The annotation is something else, because this is something hard. You need a professional to tell you, for example, what would that mean here? What is the meaning? So here you can have maybe some kind of filaments that are associated to the maybe reticulum endoplasmic, although I don't know. We need at this point a professional to look at least from what we know. But what is interesting is to discover things that we don't know, things that are not that obvious, right? But I think we can do that, because if you can start to use some kind of clustering algorithm, just for example for vesicles, and you see vesicles that are not necessarily cluster, but it appear more than than the average, maybe you can discover some kind of connectivity. Okay. Now, this was a parenthesis about this. Let's go back to functional connectivity. So the functional connectivity, they are coming from time correlation between events. So you remember this figure? This is what we discussed a few minutes ago, where you have the region of interest that are well identified, and from this region of interest, you can plot with the rectification when you remove the baseline. You can look at the specific events that are above a certain threshold. For example, this one you don't care, but this one we care. And then you look in your distribution, when is going to be the next event? So the next event would be this one, and there's a third one which comes from from one of these guys, and so on. So you can repeat. You can order your time series iteratively. Once you have found this one becomes the first one, and you look for the next one, and so on. By looking at this, you can generate, when you go from the first to the second, a link between the first to the second. And you say that there is a potential link. And iteratively, when you do this, so you look at your different time series. From the first two, you move to the next second and the third one, and so on. And so you add your path now between the second and the third, the same way you added the path between the first and the second, and so on. So you do that iteratively. And when you segment according to the minimum, here is the minimum on this not corrected baseline, you get a succession of events between the dashed line here for this different subperiod that leads to some paths. It's a way to segment a path. Because when we say go to the minimum, maybe that's it, the activity stopped, and then it resumes again when you start these new epochs. All right, so for each subperiod, as I described to you, you generate one pass, a second pass, a third, and so on. And so, since your astrocyte are fixed, they are the nodes of a graph. And you can estimate the number of times you have been getting a relation between two, and so you increase your connectivity index by this, right? And so you can construct a matrix. You have 20 astrocytes, so you have a matrix 20 by 20, and each time during your acquisition, each time you have a pass you add between neuron I and neuron J, you can add, so hope it works, yeah. So between neuron I and J, you have your connectivity matrix. Each time you add a point, you have +1 into this, so in the end you get a number, let's say 50, and so on, and then in the end you get a matrix that will represent your graph, your ensemble of connectivity, so you know where are the nodes, and you can use this matrix to extract properties. Okay, so this is what we have been doing. So we looked at this reconstruction across 3 regions of the brain: Cortex, Hippocampus, and a region called VLPO. And so, just by eye, for about the same, so all the movies are acquired, I think, during 5 minutes, so you have the same standardized protocol, so to make sure that you don't have longer movie, one compared to the other. And what you see here is the results. So this suggested that you had difference in the connectivity, in functional connectivity for these different regions. One was more or less homogeneous, but this one you could see there is a red part and there is a yellow part, suggesting that there are subgroups that were more preferentially activated. And similar for the VLPO, it was in between the cortex and the hippocampus. All right, so now that you have this representation, you would like to go to quantification. So this is the pairwise correlation matrix, the one I just wrote to you. So you look as a function, so you can normalize from 0 to 1. Let's say the maximum is 50, you normalize by 50. And you can look at the hot color. Hot color means interactions. So you can see where you have interaction and where you don't have interaction. And then there is a method that we developed at that time is to put a threshold, saying, suppose you threshold the one that have a little amount of connections. How many connected neurons remains? How many connections remains? And you do this process from removing those who have little connections to all the connections. So at the end, there is nothing left. And if you do this process, we said that if you do that across the 3 regions, maybe we would have a way to better quantify the level of connectivity, because as you remove connections, it will reveal the statistics of those that remains connected. And that's what we found. So you do that over a certain number of experiments, and this is the mean and this is standard deviation, and this shows that the level of connectivity is different in the 3 regions, having the one that are the most stable, which is the one of the hippocampus that that stay, as much as they can when you increase this threshold. So this was the graph that we proposed as a way to measure this level of connectivity across these threshold experiments. All right, and then we started to look at the statistical properties. So for example, we ask, are there astrocytes, that is astrocytes that are heavily connected to most of the one in the network? So this is the distribution of the connectivity. Yeah, maybe I should have presented the degree first. Yeah, let's start with the low degree because it's... So you look at the degree. The degree is the number of link you have between the other astrocytes. And so when we looked at their distribution, the cortex versus the hippocampus, the Ca1 region, that's what you see. So you see that the Ca1, they have much more, the distribution of astrocytes that are heavily connected is very different from what you see in the cortex. So the sparsity analysis was what you get in the matrix. We look at what you get here. So the sparsity was how many zero you have in the matrix. You look at Cij. Some the coefficient can be zero, some are not zero. And so here we saw some differences between this region which shows some heavy connections. Again, this is to quantify the difference of the connectivity that is coming from this time series analysis. Yeah, so this is now the transition probability. The transition probability matrix is obtained from the Cij matrix by normalizing so that it becomes a probability matrix. So how do you do this? You take your matrix, Cij, and you normalize it, Cij, over all the rows. So you have to sum on J. So that when you sum this, it becomes equal to 1 over the rows. So it becomes a probability matrix. And from this, you can use this, for example, to study transition probability. This has been done quite a lot in queuing theory in the 80s, or even before. So, yeah, so this is the age weight. So okay, this is another statistics. So the transition probability matrix looks like this. The age weight is, instead of, you just look at the distribution of Cij here. Another kind of statistic that we develop here is the path duration. So if you have a given path or length, it's the duration is how long it takes to move on into a path that you have. And this has been fitted by this curve. There is no specific reason for this fitting. It was just a way to quantify the difference. Okay, so maybe I step. Yeah, the past step. This is also what I went a bit quick on this. So this was the hub astrocytes, the astrocytes that are well connected, and I think what we did here is this violin plot about this hub astrocyte, showing the difference between the two regions. So here, obviously, there are much more connected astrocytes in the hippocampus than in the cortex. Again, we haven't correlated this with anatomical connection. It just comes from functional connectivity. And here, this was the past number of steps in the past. You see here some differences with this, the peak here again in Ca1, suggesting that there was maybe some differences. I think we quantified the difference between the graph here, which was significant, especially because of this. All right. Is this what we have been doing okay. How do we know that if you add more data, that all what I presented to you will survive? Is going to survive or not? For this, you need to introduce a notion of convergence. That is, if you add data, if when you add your data, for example, you look at one point and a node, let's say an astrocyte, and you look at the connectivity, that is a number of astrocytes that are going to be connected to this one. If after some amount of data, let's say the number remains 3, then you are confident that, if you increase and it saturates at one number, that something is stable. So you need to introduce somehow briefly a notion of convergence that we try to implement here with the small amount of data that we have. So suppose you have a data d1, dn. So this is, for example, the sub-period that you segment. You have 10 sub-period, 20, whatever. You have a certain number of them. This is what you get. And you can get more of this. And you don't have necessarily to consider this one first. You can also make what is called a permutation. That is, I can consider another one. Instead of d1, d2, d3, I can consider d3, d2, and whatever. A permutation. So that if I reconstruct from this ensemble of data, or that ensemble of data, in some sense, I wish at some point to converge. I wish at some point to say, look, if I can take a certain fraction of it, another fraction of it, it's going to give you the same results. So, to formalize this, we say that if I have a data or permutation of the data, I can reconstruct the matrix, let's say the connectivity matrix from this ensemble. And the connectivity matrix, so this is, I have n data sets, and I will look at what brings the next data sets. And for example, I can look at the number, if I get yes or no, a connectivity from, let's say, I and j, from this process. So it's a way to increase or to update one of your statistics, connectivity matrix, whatever you have, whatever statistics you are interested in. So you increase. Great. And the question is what you are expecting when you get a large number of these data sets. So you increase, you have your parameter, the value of this parameter can depend on the permutation that you have, sigma, that you compute from your matrix here, and you are interested to make sure that this has a limit. That is, when you have a certain number of n and n tends to infinity, whatever it means in reality, you have enough data, that's what it means. You want to have a limit of this. You want to make sure that it converges. But this should be independent of the permutation. So, in that sense, if you have this, it should be independent here of the permutation that you have on your data, right? This is independent of sigma. So again, I have this limit. Could increase, so you have your reconstruction matrix with whatever property. For example, in this case, you have the degree that I illustrated here, I. And so the convergence parameter, you have a degree of a node, you have the mean and the variance, and the limit, as the limit n tends to infinity, if you have, let's say, a certain number of, let's say 3 here, this will be your degree at infinity when n is large. which means that in some sense it has converged. The same if you want to have the hub neurons, that is, you look at the degree that are above a certain quantity, this will be your hub. And it means that if you add more data, it doesn't matter. It does not matter. So this is the notion of data convergence. When you add more data after some n, it should saturate, it should not move, and this construction should be independent of sigma, of your permutation. So because we had, so how do you implement this? Because this will be a sort of proof. Because if I give you the reconstruction that I've done so far, you can ask, are you satisfied with this or not? Are you going to stop here, or do you need more data? So that's what we did. Yeah, I don't know why I'm showing you the connectivity matrix. Can't remember. Okay, so that's the connectivity matrix. I don't. Yeah, sorry, I'm not sure why it is here. Okay, so what we did is this. We said, let's split the data. Let's split the data set into two sets, N1 and N2. And I'll start calculating from N1. Let's say, suppose you have 70% of the data, and it remains 30%. So that you implement a convergence approach. Convergence means you want to see what happens when you are going to add more data. So I start with 70%, and I start to increase. I want to study the convergence of a chosen variable. So for example, you ask about the degree. Let's choose the degree that you have constructed up to N1 and see how the degree is going to evolve when you add one more of the data sets. Right? You can choose whatever you want. Mean and variance of the graph node degree, mean pass length, number of hub astrocyte, whatever active area per subsection. At this stage, what we did also is we said what is the area. We can quantify the area where we had a given pass. Number of active rows, okay, whatever you want. Check for little variation when approaching n. So now, since you don't know what is the limit, right? Maybe it's going to converge, but if it converges and you see some kind of saturation, that's good, because it means that you're approaching a limit. You don't know what is this limit, but you're approaching this limit. So check for little variation when approaching n. Check that the convergence is similar when changing the order, which is the permutation. So we started with, we had n equals 20, we started at 5, and that's the result. So, for example, let's go to the mean pass length. It's the most clear. So the color means the different, we have the two regions. And so we started here, so you normalize the length, and we added things. And what we saw is after 10, there was little changes. Okay, so we saw it, for different things. For example, the node degree here, we had, this kind of behavior. It seems to be one was, the CA1 was not changing, whilst the other one was converging, because node degree, this is it quantifies in integer, so this was quite okay. Astrocyte, you can see, how things are converging, and we were quite satisfied to see that the elements we were calculating were converging. So you could say, look, I can see there's a little trend of going up. That's true, but at least now you have the methods to justify how much of data you need to reconstruct. And obviously, if you were here at 5, this will not satisfy. But when you see this convergence, you could say, look, after 10, 12, now I'm confident that I've reached some kind of stationarity. Okay, so this is something that we put in that paper that was not much discussed in the literature about this convergence, and that gave us confidence about the methods and the amount of data we were using, and that what we had was acceptable. Okay, one more piece of information, and we will stop here. Maybe you can say, look, what you are doing is specific to a moment of time. Maybe if you repeat the experiments in the future, it will give different results, which in that case you're in trouble. Because if it's the case, then what you have been doing captures something that is transient, or you don't know exactly what it captures. So then we move to something called time-lapse analysis. We said, okay, let's record at a time t1. Let's apply all the pipeline to time t1. Let's record later in the future at time t2 without mixing the data. And do we get the same reconstruction matrix? So that's what we did. And so here we found, so that are the two graphs that are reconstructed at two distant moments of time. So the highly connected node was 78, this is 75. Look, the matrix looks quite similar. This is what you get here. And we started to quantify what you get between time t1 and time t2. And basically, in terms of connectivity, in terms of the strengths, we're quite happy that everything was quite intermingled. Okay, there were some changes, but overall, the correlation threshold and the behavior was quite similar. So again, we said that, what we quantified it. We show that there is no significant changes here. It's what has been quantified. So here it's not significant. So this shows that the construction we made is stable over time, which is important because it says that the snapshot you got and the reconstruction you made captures something that is stable, right? And so that was one of the novelty here of the study, and I think it's important in future study of this reconstruction to have these two type of verification. That one, you need to have stability of when you increase the data sets, and also if you have two timescale, sometimes it's not possible, but when it's possible, to have to show that you can have a similar reconstruction. Okay, so again, those are proofs of concept that we brought because of the problem that we were addressing. All right, we finish with a series of open questions. The notion of accuracy of reconstruction. How accurate are we? I think there is not much math on this. So again, this talk, this class today, I didn't put much mathematics. I think the mass of graph theory is well known, but the graph, the theory behind here is still to be developed. So the accuracy of the reconstruction, this needs to be understood. How can you measure the accuracy? How do you compare? What do you compare with? This is not clear. Comparison functional and anatomical. You have anatomical, as I showed to you, connectivity. You have functional connectivity. How do you compare? This is not easy because how do you record at the same time the anatomy and the functional connectivity? It should be doable. I'm sure people, the Allen Institute and other are working on this. How to relate the number of synapses for example, in the case of neurons, how to relate their protrusion, the way protrusion are orchestrated in the case of glial cells, to the fact that you see indeed change in the calcium. So there are various methods of reconstruction. For the astrocyte, I think we are the first one. This was the first construction we have. There are many methods on neurons. There is no general consensus about which one is better. I mean, you can see sometimes there's improvement there, improvement in one, in the other, depending on the context. It's still unclear today if what you are reconstructing, and that's a project that we are interested in, how this is associated. So you construct network, but you can reconstruct paths. Are these paths associated with cognition, with a task? That is, if you have a task or if you have social interaction, do they have the same behavior again and again and again, or are there viability? And then how do you measure viability? Do you have to associate a graph? Do you have to associate an ensemble of paths? Do you have to associate statistics of these paths? That's what we are working on with Philippe. Yeah, and mathematical theory to be made about, how good is your reconstruction, especially when you are going to get not one network, but when you are going to get millions of network. If I give you millions of network, millions of paths, how do you identify the relationship between the paths? And based on this, maybe this is a way to go to coding. Coding information was, the paradigm was, look at the spikes. Spiking was the paradigm. But maybe we've moved on to a next generation of understanding coding through the paths, through the local graph network that represents something, where maybe there's something encoded, and how the path is designed tell us how the information is retrieved or something is calculated. That's where we are today. So coding by just looking at raster plot, from what we have understood up to now, this is probably not enough. We need to go through this path, identify this neuronal ensemble or glial ensemble, or the interaction between the two, and try to identify structure. Probably we should use machine learning in time-dependent manner between millions or large ensemble of graphs. Maybe those graphs have some kind of universal structure. All of this has to be discovered, and this is where I think the research is today on this. So that ends the class about reconstruction. I wanted to have one more class about neuronal reconstruction, but since the paper is not out, I think this can wait next year. So next time I'll ask Thibaut to come to give you a class about spatial statistics, and spatial statistics apply to glial cells, and I think we have more or less covered all the curriculum of the class. Okay, so are there questions? All right, so let's stop here.